In the vast expanse of the ocean, where the waves caress the shore and secrets hide beneath the surface, a remarkable discovery awaited the world of science. It was a discovery born out of necessity, driven by the need to ensure the safety and effectiveness of medical equipment and vaccines. In the not-so-distant past, the detection of bacterial contamination posed a significant challenge in the biomedical field. The methods available were time-consuming, expensive, and ethically problematic. Scientists and researchers tirelessly sought a better solution, a way to protect patients from harmful bacteria while minimizing the impact on animal lives. Amidst this quest, a group of dedicated scientists set their sights on a unique marine creature, the horseshoe crab. These ancient arthropods possessed a fascinating defense mechanism, a natural armor against bacterial infections. The scientists wondered if this defense mechanism could hold the key to solving the contamination puzzle. Carefully, the scientists embarked on a journey to study these enigmatic creatures, venturing into the depths where horseshoe crabs roamed. They observed and marveled at the crab's ability to clot their blood in the presence of bacterial endotoxins, a telltale sign of contamination. Excitement stirred within their hearts, for they knew they had found something truly remarkable. The scientists harvested a small amount of blood from the horseshoe crabs, ensuring minimal harm to the creatures. Back in their laboratories, they meticulously separated the plasma from the amoebocytes, the specialized cells responsible for the clotting mechanism. These amoebocytes held the key, the vital compound that would transform contamination testing forever. Through delicate processes, the amoebocytes were lysed, releasing a plethora of enzymes and proteins. Among these miraculous substances was limulus amoebocyte lysate, affectionately known as LOL. The scientists freeze-dried and processed LOL into a powdered or liquid form, ready to be utilized by the biomedical industry. As word spread of this groundbreaking discovery, the demand for LOL grew exponentially. It swiftly became an indispensable tool in the fight against bacterial contamination. The pharmaceutical industry embraced LOL, utilizing it to test the safety of a wide range of medical equipment, from surgical instruments to intricate implants. The reliability of LOL testing brought peace of mind, ensuring that these vital tools would not harbor harmful bacteria. But LOL's impact extended far beyond medical equipment. In the realm of vaccines, where the health and well-being of countless individuals hung in the balance, LOL became an unwavering guardian. Vaccines, the life-saving elixirs, were subjected to rigorous quality control measures. LOL played a vital role in this process, assuring that vaccines remained pure and free from bacterial contamination. The test itself was elegant in its simplicity. A small sample from the medical equipment or vaccine would be mixed with LOL. The scientists would then observe, their eyes locked in anticipation, for the telltale signs of clotting. If the sample clotted, it signaled the presence of bacterial endotoxins, prompting further investigation and safeguards. Limulus amoebocyte lysate had transformed the biomedical industry. It offered unparalleled sensitivity, capable of detecting even the tiniest traces of contamination. Patients could rest assured, knowing that the medical equipment used during their treatments and the vaccines administered to safeguard their health had undergone rigorous testing, all thanks to the incredible power of LOL. Yet, as the story of LOL unfolded, so did the awareness of the importance of conservation. The horseshoe crab, a creature that had unlocked the secrets of bacterial contamination testing, faced its own struggles. Habitat loss and overharvesting threatened their very existence. Conservation efforts emerged, with regulations implemented to protect the horseshoe crab populations. Scientists and researchers tirelessly worked to develop synthetic alternatives to LOL, striving to reduce the reliance on horseshoe crab blood for production. These efforts aimed to maintain the integrity of bacterial contamination testing while minimizing the impact on these ancient creatures. And so, the tale of Limulus amoebocyte lysate continued, weaving its way through the intricate web of science and human ingenuity. It stood as a testament to mankind's unwavering quest for knowledge, the pursuit of safer medical practices, and the delicate balance between progress and preservation.